Let's start with the agenda really quick. We're going to have an overview of the technology related to CytoViva's Darkfield hyperspectral imaging, and we're going to break that down into the microscopy piece of this, and then talk about how hyperspectral imaging is integrated onto the microscope technology, and then actually show a live image of what this looks like as a finished product. And then after we do that, we're going to take you guys through a number of applications. We're going to look at plasmonic nanoparticles that are used as biosensors or used as drug delivery vectors. We're going to focus on liposome and polymer nanoparticles, which are very robust, applications surrounding those nanoparticles as drug delivery vectors. And we're even going to cover biological nanoparticles, and we're going to look at viral particles in a biological matrix, just to give you a sense of everything from plasmonic down to pure biological particles, and then we'll have the Q&A as we mentioned. So again, thanks for joining us today. Want to just give you a brief overview about who CytoViva is. Interesting enough, we are born out of academic research at Auburn University, which is a state university, a very large state university that's located outside of Atlanta, Georgia, in the southeastern United States. Our company was formed specifically to commercialize label-free nanoscale imaging and analysis that had been developed at Auburn. And we're located in the research park at Auburn University, and we leverage the university quite heavily to help us in our ongoing development of these applications that we provide to the greater research market around the world. And to that end, this technology that we're going to be demonstrating today is currently utilized with a lot of ubiquity around the world. Currently, there are over 400 research labs that utilize CytoViva technology in every continent around the world, every inhabited continent around the world, I guess I should say. There's not one in Antarctica. And it's heavily referenced. We're up to almost 2,000 references in journals that feature data and images from this technology. So we're starting to see rapid and major adoption. Rapid and major adoption is occurring or has occurred primarily in laboratories around the world that are involved in nanomaterials development and synthesis, nanodrug delivery, nanobiosensors, nanotoxicology related work. And so this slide is just designed just to kind of give you a sense of the types of labs that have acquired the technology and the types of work that they're doing with it. We have a number of systems that are deployed at the Food and Drug Administration in the US and other FDA-oriented facilities around the world, where a lot of nanodrug delivery testing is ongoing, looking at efficacy of nanodrug delivery vectors, but also nanobiosensors as well. And in the early days, I would say that the majority of the nanolabs were involved in nanotoxicology. I show Adolf Merkel Institute as an example of that has been involved heavily and still is in environmental nanotoxicology work, looking at a wide range of nanomaterials and their impact in a wide range of environmental conditions, all the way from whole animal organisms to plants, water, etc. I want to quickly show you what the system looks like before we begin. So this is the footprint of a hyperspectral microscope, and it has some very unique elements to it that I want to point out. First of all, you see that everything is centered around a very basic research-grade optical microscope. Typically, when CytoViva provides a full system to a research lab, we typically do that on an Olympus microscope frame. We have the ability to integrate Darkfield hyperspectral imaging on other microscope frames and can sometimes do that on existing microscope systems that may be currently in a lab. But the microscope covers everything. The system covers everything from the light source to special optics that CytoViva provides that we're going to talk about, specialized objectives, a a translational automated stage that's used for the line scan hyperspectral image, where we build hyperspectral images pixel row by pixel row in a push broom or line scan method that we'll talk about a little bit. An integrated spectrograph and detector for the spectrophotometer system that is then connected into very robust hyperspectral image capture and image analysis software that allows you the ability to capture dark-field hyperspectral images of nanomaterials in a wide range of applications, a wide range of environments, of biological environments, and then conduct analysis on those. Now, the microscope itself can be configured for whatever application the research calls for. And in most biological applications, we work in either transmitted bright field, but more likely than not, we're for nano, always in transmitted enhanced dark field. For material science applications, or even for biology applications where the substrate may be opaque, we can work in reflected mode as well, from the top down on the microscope onto the sample. We can also work in fluorescent microscopy mode, and there's some very interesting things that we can do there to be able to spectrally measure fluorophores in a wide range of biological environments if necessary. And the hyperspectral imaging can work in the visible near infrared from 400 to 1,000 nanometers, or in the shortwave infrared from 900 to 1,700 nanometers across those wavelength ranges. And we'll talk about that a little bit more. But what I want to do first, before we do anything, is I want to actually show you what a dark field hyperspectral image looks like and the types of analysis that can be conducted with it. And after we do this, we'll transition back into the technology to talk a little bit more about how this happens. But what you see on my computer screen here today are HeLa cells that have been incubated with gold nanoparticles that have a targeting protein for uptake of the gold particles into the HeLa cells. And we refer to this as a data cube, and it's really a 3D image. So the X and Y axis of this 3D image are spatial, and it's a two-dimensional spatial image. The third dimension we typically think about as being spatial, but in this case, the third dimension is spectral. And what we mean by that is behind every pixel, as you see in this zoom image here, there is spectral data, the third dimension being spectral data behind the pixel. And if I right-click the Z profile, the Z axis being the spectral third dimension, right, produces a spectrum. And you see the spectrum of this area here. Now, these gold things that we see in the tissue are, in fact, gold nanoparticles that have been internalized by the tissue. And we know they're in the tissue based on the spectrum that we see, where we see a pretty broad full width half max compared to if we were just looking at this nanoparticle in solution. But also, if we were to focus up and down in the spatial Z-axis, with the microscope, you would see nanoparticles appear and disappear in different focal planes of these cells. We're on a specific focal plane where the nanoparticles appear the brightest in this image. That's why they're so visible. If I want to collect spectrum from here, I can collect spectrum. And if I come over to a tissue area where there is no nanoparticles, you can now see I have two spectrum. The first spectrum is the spectrum of the gold particle in the cell. The second is the spectrum of the cell itself. So you see a couple of things. You see a shift in the spectrum. You see a broadening in the spectrum from the nanoparticle to the cell only. You also see significantly more reflectance spectral intensity being measured of the gold particle, because gold has more spectral reflectance characteristics in its spectrum than the tissue does. So based on this information from this dark field hyperspectral image of gold nanoparticles in HeLa cells, we're able to determine that gold particles are present. we're able to get information about where the gold particles are in the cells and the relative amount of gold particles that may be present in the cell vis-a-vis this sample image versus other sample images where you may be using different concentrations of gold, targeting proteins, or different exposure timeframes to measure those differences and how they're reflected in the uptake. Now, it's important to mention that these cells are label-free. This is the endogenous scattering that we see from the cells based on their natural scattering tendency from the material of the cell. Also the same thing in the gold, no fluorescent labeling. So we talk about label-free, and this is what we're talking about. One of the things you can see if we focus on the cell here where my cursor is, we're looking at, I'm outlining the nuclear membrane of this cell and the nucleus here, and outside of that would be the I'll call it the cytoplasmic, the non-nuclear area of the cell. And if we look across these cells, we can see that nanoparticles seem to be mostly aggregating, probably in cell vacuoles outside of the nucleus, not crossing the nuclear membrane, which makes sense. These nanoparticles, before they were taken up by the cell, were around 40 nanometers in size. That's too big alone to generally cross the nuclear membrane. And so we can observe that qualitatively as we see here. But based on the unique optical spectrum that we see here in the white spectrum from the nanoparticle in the cell, we can apply a spectral mapping algorithm to this hyperspectral image that's built into the hyperspectral image analysis software to be able to spectrally map that spectrum. And that is the spectrum of the gold in the cells. And that spectrum was built into a spectral library. And then it was filtered against negative control cells to ensure that we have no false positive image. And then when I turn on the areas that are not mapped, you can see where the nanoparticles are mapping in the cells. I turn it on and off to give us quantitative confirmation that we have nanoparticles, relatively how many nanoparticles we have, or the amount of nanoparticles that we have per cell, and where they're co-localized in the cell. So this is really critical information as it relates to nanobiosensor-related research, nanodrug delivery-related research, et cetera, even nanotoxicology as well. So I wanted to show that live image, so to speak, of nanoparticles in cells to really give you a sense early on of the capabilities of the instrument. With that, I want to transition now back into the actual presentation, and we're going to talk about how it is that we're able to build that image, create that image, and acquire that data. And let's first talk about the patented enhanced Darkfield microscope optics that CytoViva develops and integrates onto, in this case, that we showed you, the Olympus microscope to enable label-free nanoscale imaging. These are the condenser optics, we'll call them, that fit into the condenser mount of the optical microscope. And they're designed to create a very high signal-to-noise ratio image. And we do that by managing the light from the source illumination very precisely to a cardioid oil condenser Darkfield system. And the light comes in at a very shallow or oblique angle such that the source light itself has an angle that's much greater than the angle of light acceptance, the numerical aperture of the objective. So that when light interacts with the sample on the slide, scatter from that sample goes into the objective, but no interference, which is always going to be the light source, is in the field of view. And that's why we were able to see this really crisp image of cells and nanoparticles in the Darkfield hyperspectral image that I just showed you a little while ago. And everywhere there wasn't cell and everywhere there wasn't nanoparticles, it was dark. It was black. And that's how Darkfield actually works. Now, CytoViva's Darkfield optics are very different from any other Darkfield optics that are available. Most of the major microscope companies, Olympus, Zeiss, Nikon, Leica, provide Darkfield optics. But CytoViva has a patent on its enhanced Darkfield optics that serve to manage the light much more precisely from the light source to the condenser that produce this 10x increase in signal to noise that we're referring to. And rather than go into the physics of how that works, let's just show you some data on that. This is a paper that was produced a number of years ago by a research group in Korea that had acquired these Darkfield optics, these enhanced Darkfield optics, to do microchip electrophoresis work. They had been for years using standard Olympus Darkfield optics, and they were able to get images such as this of these 160 nanometer iron oxide nanoparticles. By using the same microscope and just upgrading to the CytoViva enhanced Darkfield optics, they saw a dramatic improvement in signal to noise ratio, the ability to detect the scatter of the nanoparticles, and to be able to measure that. And when they measured that signal to noise ratio, as you see here, they measured about a 10x increase in signal to noise ratio. So CytoViva first provides an ability to quickly and easily detect scatter at the nanoscale of nanoparticles, viral particles, cells, subcellular images, even tissue that you might want to look at. And that's really important because if we're going to capture a hyperspectral image, and you guys saw earlier that the hyperspectral image produces a spectral response measurement in every pixel of the high resolution image. I showed you that a little bit earlier. That we're able to measure and analyze that data. As I mentioned, we capture, and in most nanobio applications, we work in the visible near infrared from 400 to 1,000 nanometers. However, there are some applications, for example, there are groups that are developing nanorods that produce their primary spectrum in the shortwave infrared, 900 to 1,700 nanometers. And they're doing that principally when they're developing nanorods as biosensors to get away from any autofluorescence, which may be in the tissue. So we have the capability of measuring optical spectrum either in the visible near infrared or the shortwave infrared. When we capture a hyperspectral image, we're capturing that at pixel sizes that can be as small as almost 100 nanometers and larger in the image. The image that we showed you was approximately 200 nanometer pixel sizes that we were looking at a little earlier. Now, the spectral resolution, if we capture the full spectrum from 400 nanometers to 1,000 nanometers in every pixel of the image, we do so at very high spectral resolution. Spectral resolution in the system is approximately 2 nanometers. And what that means practically to you is if you were looking at a nanoparticle and that same nanoparticle with a functional group on it, maybe that's a drug load or something else that may be added to the nanoparticle. If the functional group slightly changed the peak from, say, 450 to 455, or it caused an absorption from 450 to 455, you could measure that 5 nanometer shift with great accuracy, given that you have 2 nanometers of spectral resolution. However, the most interesting thing about a hyperspectral image is that all of the spectral data is presented as a spectral curve within a color image. The hyperspectral image capture and analysis software is designed to provide you the full spatial context of the spectral data, why we call it a hyperspectral image. And the image that we showed you of the HeLa cells in the gold is almost identical to the image you would see with your eyes on the oculars or a color optical camera image that you would capture. But the beautiful thing is that there's all kinds of detailed quantitative analysis that you can do to look at differences in the spectrum between, for example, nanoparticles that are functionalized versus those that are not, aggregating versus dispersed nanoparticles in a lot of cases. Nanoparticles that are in different areas of the cell can provide different spectral qualities that can be measured and mapped, for example. If you're going to do hyperspectral imaging of anything that's at the nanoscale or close to it, having the darkfield optics that produce the really high signal to noise ratio is critical. And we're just showing that as a great example here. This is a single 100 nanometer gold particle. We captured a hyperspectral image of it, again, using standard off-the-shelf optics, I believe, from Olympus. And with the translational stage, we can go back to the same XY registration, excuse me, where we capture a hyperspectral image with the same exact settings, only using the enhanced darkfield optics on the right from CytoViva. The signal to noise ratio is intuitively obvious. And these are 100 nanometer particles, or a 100 nanometer particle, which shows you how you can get down below 100 nanometers quite well. We have the ability with plasmonic nanoparticles to detect scatter down around 15 to 20 nanometers quite easily. Polymer nanoparticles or metal oxide nanoparticles in the 20 to 30 nanometer range, polymer nanoparticles in the 30 to 50 nanometer range, lipids down to the 60 nanometer range, 70 nanometer range. Different nanomaterials produce scatter in more and less efficient manners as I've gone up the scale there, but that gives you some sense of the expectation of the detection limits that we can do. With that, we're going to transition away from how the system works, and we're going to talk a little bit about some application examples. We mentioned earlier that we placed almost 400 systems around the world, and I would say the vast majority of those are in laboratories that are working at the nanoscale, and many of them that are working at the nanobio interface. So that's what we're going to focus on today. The first application that we're going to focus on are confirming silver nanoparticle dispersion and aggregation. This was a group that was developing ionic solutions that will allow these nanoparticles to remain nicely dispersed in this media over long periods of time relative to a wide range of applications. And so if we capture an image of these silver nanoparticles, which were originally 30 nanometers based on manufacturers' measurements of these, we see that when they're in water, we visualize lots of aggregation. Silver nanoparticles in the 30 nanometer size, when they're truly dispersed, they produce a reflectance spectrum in the 450 nanometer range. And so they are blue in nature. If you just look at them in the eyepiece here, here's their spectrum when they're nicely dispersed. And you see how much homogeneity we have in the spectrum across these nanoparticles when we look at them in the nicely dispersed in the ionic dispersing solution. When we look at them in water, however, though, we see that the nanoparticles are aggregating. As plasmonic nanoparticles aggregate or get bigger, they redshift. And so you can see the very smallest particles here appear blue, but you also see green, yellow, red as you see aggregation of these nanoparticles, and likely some oxidation of these nanoparticles are occurring as well in the water media as opposed to the ionic solution. And also, if you measure the spectrum of these individual areas of nanoparticles, you see they're very heterogeneous in their spectrum. And that's because of the aggregation that we're looking at, not only in their peak and the width of the peak, but also in the peak wavelength redshifting as well. So they're not very homogeneous in water. They're super homogeneous in this ionic solution that's designed to keep them highly stable and highly dispersed. So this is an example of an application to just look at the nanoparticles in solution and understanding the characteristics of those nanoparticles based on both observation and spectral measurement capabilities. Now we're going to look at plasmonic nanoparticles in cells and tissue, and this is going to be similar to the live hyperspectral image that I showed earlier. Here we're looking at cells that have taken up gold nanoparticles in the cells. And like last time, we see a nuclear membrane here, and we see nanoparticles that have mostly been internalized in, again, what I'll call the non-nuclear cytoplasmic area of the cell. If we look at those identical cells that have not been exposed to nanoparticles, wow, it's intuitively obvious just from a casual observation that the cells with nanoparticles, easily you can see them present, especially when compared to the cells without nanoparticles. However, if we conduct the spectral mapping of that, we're able to quantify that again. And so, for example, if you're trying to develop nanoparticles for photothermal therapy applications, which is what I believe these nanoparticles were being developed for in this application, you want to understand, based on different targeting proteins and different exposure times and different exposure amounts, what the nanoparticle uptake in the cells are so that you can optimize that for your photothermal therapy application. This technology allows you to do that in a quantitative way and a super high qualitative way as well, with both observation and with spectral measurement capabilities in the cell. That can transition from mammalian cell culture to ex vivo as well. Here we're looking at gold nanoparticles in pancreatic tissue. There's obvious areas of aggregation of the particles in the pancreatic tissue. And then there's some areas where it's dispersed, and you might have to rely on the spectral signature to help you really see where the nanoparticles are. By applying the spectral mapping algorithm, though, we're able to do that. There are areas that we're spectrally mapping where it's not super easily visible that nanoparticles are present. Also, you can use a class distribution out of this software that will tell you the amount of spatial area where you're mapping for nanoparticles. Now, that's not a volumetric readout of the total amount of nanoparticles in the total amount of tissue, but it gives you the total amount of spatial area per pixel covered by nanoparticles versus those that are in tissue, okay? We're going to now look at some drug-loaded liposomes. As most of you know that are involved in nano-related applications, that liposomes are probably the most well-received nanoparticles for drug delivery by the regulatory agencies such as the Food and Drug Administration and other types of agencies because of their clear biocompatibility and other related benefits. Here we're looking at doxorubicin chemotherapy-loaded liposomes, and we're just looking at their optical spectrum on a glass slide. This spectrum is very interesting. Doxorubicin, as many of you may know, has a natural fluorescence. It has a peak, if you measure it, somewhere around 575 nanometers. If you look at these doxorubicin-loaded lipid liposomes, you see that it has a real interesting sharp peak exactly where the doxorubicin natural fluorescence is. That's part of this overall spectrum. And that can inform us to be able to identify where these dox-loaded liposomes are in this mammalian cell culture. You can't really tell the difference between subcellular data and such as endosomes in the cell versus liposomes. That's one of the reasons why liposomes are so well received, is that they are so biocompatible and work so well in that regard. So if you look at a control cell versus a cell with liposomes, you may be able to see some differences, but it's really qualitative in nature. But by applying the spectrum of the liposome with dox, you can spectrally map that with a high degree of quantitative confidence to understand where these liposomes may be in this cell at the time that the image was captured, okay? Second only to liposomes probably are polymer nanoparticles for their biocompatibility. What we want to show here is the difference between a polymer nanoparticle without a drug load, we'll call that the vector control, versus polymer nanoparticles with the drug load, the active pharmaceutical ingredient loaded on the polymer nanoparticles. In this case, the API, or the active pharmaceutical ingredient, is cisplatin, which is another small molecule chemotherapy drug, which is very well understood, approved by most regulatory authorities. In this case, the group is trying to label the polymer nanoparticles on the surface very effectively with the cisplatin. Now, the cisplatin can dramatically change the way that the polymer nanoparticle absorbs and reflects light. And with hyperspectral imaging, we can measure that very effectively. This is the nanoparticle control without the cisplatin, and you can see how dramatically the spectrum of the polymer nanoparticle changes as a result of the cisplatin small molecule drug being located on the surface. And by applying the spectral mapping algorithm, we're able to demonstrate that every polymer particle in this field of view contains cisplatin on the outside of the particle. So remember, this is a two-dimensional spatial image of three-dimensional objects. These polymer nanoparticles are spheres. So we're looking at basically a cross-section of them. And the fact that we're mapping the API, the cisplatin, on the outside of the particles indicates that it's on the outside, not on the inside. Now, we have looked at, for example, drug loads in the bilayer of lipids, and you'll see the drug load in the bilayer of the lipid in that application, for example, when you conduct this type of analysis. But this is an application just demonstrating how well you can look at functionalization of individual nanoparticles. And this is very important to regulatory authorities. Oftentimes they get ICP mass spec data that gives them very accurate quantitative volumetric data sets on the active pharmaceutical ingredient and the vector. But they also want to know, okay, how are those two things interacting with each other? How stable is the API with the vector? Darkfield hyperspectral imaging gives you that information in a highly visible way that helps to cross-correlate these destructive quantitative techniques such as ICP mass spec, and can be very helpful in that regard. Finally, we're going to look at a very unique application. We're going to look at actual viral particles in whole blood. This is just to show a very unique application of use of the technology that probably doesn't have a lot of application or utility for most of you that are engineering nanoparticles for a wide range of applications, but it's interesting to see. We're looking at virus particles that are in whole blood. Those circles that you see are 7 to 9 micron in diameter red blood cells. The little blue guys that you see in here are viral particles that are anywhere from, say, 100 to 150 nanometers in size. You see a lot of differences between the viral particles and the red blood cells, right? So these viral particles are nanoparticles, right? They're just natural nanoparticles as such. By using the spectral mapping algorithm, we're able to map everywhere the viral nanoparticles are in the whole blood sample. Now, these could be engineered nanoparticles that we're looking at in this whole blood sample, for example. But by showing the ability to see, natural biological nanoscale phenomenon in the blood, we believe it just shows the power of the instrument quite well. And we wanted to actually end this webinar by demonstrating this capability of looking at nanoscale viral particles in whole blood. So in closing today, there's some tremendous benefits of this technology as an imaging technology for anyone that's working at the nanobio interface. First of all, it's label-free. And something we didn't mention that most of you may know is that it is very difficult to put an immunofluorescent label on a nanoparticle. It's difficult to conjugate that. It can change the functionality of the nanoparticle because you're adding something artificial to it that you don't want to. And most importantly, when a fluorescently labeled nanoparticle interacts with biological matrices, it's very easy for that label to come disassociated from the nanoparticle. And if you're using a fluorescent-only microscope, you may think you're seeing your nanoparticles and you're seeing disassociated fluorophores. We've seen numerous examples of that with people that we've worked with. This is completely label-free. It provides for the nanoscale detection. It's quantitative from the perspective that you can quantify that your nanoparticles are present. You can quantify functionalization, drug loads, et cetera, on nanoparticles. And relative to a lot of other techniques, it can be high throughput. These images can be captured in a minute or two. And once you have a spectral library built of your vector, whatever it is from a nanoparticle perspective and the cell you want to target it to, the analysis can be conducted in just seconds or minutes for that. So with that, Carol, I don't know if we have any questions that we might want to answer, but that concludes the formal part of the presentation today.
Thank you, Byron. I will check the Q&A. If anyone does have any questions, you can drop them in the Q&A section or in the chat. But if we don't get any, then we'll be done.
We would encourage you, if you have questions that you might want to ask, you can take a screenshot of this really quickly. You can send an email to info@cytoviva.com. If you want to talk about your specific research or you have some specific questions that you might not want to ask now. And we'll be pleased to either just reply to your email, or if more detailed information is required, we can organize a brief Zoom call with you and talk to you about your application and see if it's something that this technique could help as well.
All right. Thank you, Byron.
So do we have any questions? I guess not.
We do not.
All right. Cool. Well, everyone, we appreciate very much you coming on. And for the participants, we'll have a recording of this and try to get it up where if you want to go back and look at it and share it with colleagues, you can. But thank you very much for attending. And with that, we'll call the meeting to be. Oh, it looks like maybe there's one question here, right?
No, they.
Just a
thank you.
Okay.
You're very welcome.
Absolutely. Absolutely. All right. Thank you, everyone, and we appreciate your attendance today.
All right. Thank you.